Dear students, good morning to all and welcome to this new semester. Hope everyone is doing great. I am Dr. Nyanandra from Department of Biotechnology. I am going to teach you the course on Advanced Environmental Microbiology. So in this part of course, we are going to focus on various aspects of bioinformatics tools, data analysis methods, and their applications to environmental microbiology. So during last semester, we have discussed about various generations of sequencing technologies like first generation, second, third, and fourth generation sequencing technologies. So these third and fourth generation sequencing technologies outputs tremendous amount of data. So that we have discussed various methods, what are the library, how the libraries has been prepared, and what are the different kinds of machines and how they have evolved. So from this data, how one can analyze and what they can infer. So these days, knowledge and bioinformatics tools and various data analysis methods are very essential to handle large-scale data such as microbiome and metagenomics. And this has become very essential for researchers in microbiology and biotechnology. So the rapid advancement in these sequencing technologies and various data analysis methods to handle this large-scale data provides an exciting opportunity to explore and to understand the microbial communities from various environments like water, soil, plants, animal tissues, and even space, and many artificial space labs, spaces, like bioreactors. So this kind of analysis really helps us to know who are present and what are they doing in that community. So let's have an overall look about what is environmental microbiology and what kinds of data are available for us and how we can handle and analyze those data in this course. So coming to the course content, the course is divided into 4 sections. Section 1 deals with genome sequencing techniques. So where we are going to deal with various sequencing technologies, library preparation kits, data analysis platforms, and genome assembly and annotation. So where we are going to see and various annotation pipelines and genome assembly tools and metagenomics. So what are the preparation methods, what kind of data we can get, and how we are going to analyze that. And coming for in section 2, this deals with microbial diversity and ecology, so which is mostly about and microbiome data. And coming to the session 3, it deals with plant and host microbe interactions, so how the microorganism is been beneficial for the plant or its host. So we are going to discuss a few things about that. And microorganisms against phytopathogens, so how the beneficial microorganisms are helping the host to fight against the invading pathogens. And coming for an session 4, so this will be an hands-on experience, so where we are going to deal with an certain microbiome data, so which will be available from an source from an NCBI or from open sources. So we are going to deal with an specific microbiome data, and we are going to make an community profiling for that by using an chime tool, and we are going to use an RStudio for making an beautiful graphs with an plotting or publication ready figures by using an R tool. So this is all about an course content, so which we are going to discuss. So this will be an 4 session course. So the course evaluation method is based on 5 different categories like attendance, presentation, assignment, midterm exam, and final time exam. So each is being distributed. Attendance, presentation is carrying in 10 marks each, and assignments 20. So overall it says that 2 assignments each 10 marks, and midterm is for 20 marks, and final term is for 40 marks. So overall, this is an evaluation pattern which I am going to follow to evaluate or to graduate to grade you students. Environmental microbiology is most closely related to an microbial ecology, which comprises the study of the interaction of microorganisms within the environment like air, water, or soil. So this environmental microbiology is also defined as study of microbial interactions, microbial processes, and microbial communities in the environment. So usually this environmental microbiology deals with microbial communities that are helpful, how they are making an element cycles and biogeochemical processes, and what are the microorganisms that are being seen in extreme conditions, and what are the microorganisms that are being seen in different habitats like terrestrial microbes, aquatic microbes, or extremophils, and that lives on different organisms. So these are the various studies which deals with an environmental microbiology. So structure and activities of microbial communities, host microbial interactions, in which we are going to see about symbiosis, mutualism, commensalism, and parasitism, and microbial communities in healthy or extreme or unusual environments. So coming for a structure and activities of microbial communities, this usually deals with which phylum is rich in community and which is dominant at specific conditions and what's the role of those specific bacteria. So we are going to say that, okay, what is the structure of this particular community in this particular conditions? So what is the community which has been present in another comparative conditions? Say for an example, like I want to find out the structural communities between healthy plant and a draft condition grown plant. So we can compare that, okay, in healthy plant, in a natural soil plant, so which phylum is rich and which has been dominant, as well as in drought region, so which microbial phylum is rich and which is dominant. So and what is the role of each and every bacteria, so we can compare that. So that's in structure and activities of microbial community. And next, coming for an host microbial interactions. So here it deals with the nature of microorganisms and its interaction with its host, say like whether the microorganism exists as symbiont, mutualistic, or commensalism, or parasitic in nature. So say for symbiosis, so this is a relationship in which one organism may depend on another for its survival. Sometimes they need each other, so mainly they rely for food and nutrients. So that's in symbiosis. And mutualism. So in this type of relationship, both host and microorganisms gets benefited. Say for an example, E. coli, which synthesizes vitamin K in the intestine of host, and mutually it's get benefited by getting a nutrients from the host intestine for its own survival. So next, coming to an commensalism. So in this relationship, one organism is benefited and the other is unaffected. So there will be no harm to the host. So they bring no benefit to the host. So while the microorganisms are greatly benefited for the inhabited environment. So example is Staphylococcus on skin. And next one is of an parasitism. So parasitism, in this relationship, one organism is benefited at the expense of its host. So the host will be affected and the microorganism will survive by getting an advantage of host. And next is of an microbial communities in healthy or extreme or unusual environments. So we are going to study that, okay, which microorganisms has been seen is an terrestrial microorganisms or extremophils or aquatic organisms and all. So these are the things which deals with an environmental microbiology. Microbial ecology deals with the relationship of microorganisms with one another and with their environment. So it is distributed into three major life domains such as eukaryota, archaea, and bacteria, as well as viruses. So as we all know that these microorganisms are seen everywhere. So they are called as an omnipresent, and which in turn impact the entire biosphere. So this microbial life plays a primary role in regulating biogeochemical systems and virtually all of our planet's environments, including some of the most extreme uh, most frozen environments and acidic lakes to hydrothermal vents at the bottom of the deepest oceans, and some of the most familiar places such as human small intestines. So studying these microorganisms, the presence of these microorganisms at various conditions and various regions, or simply we can call it as of a microbial ecology, or we are going to deal with a microbial diversity. So next, coming to the application of microorganisms. So as we know that these microorganisms are seen everywhere, so definitely they are having an potential applications in every sectors like agriculture, in which it helps us to make and drought resistant plants, pesticide resistant plants, so many, and the energy making and biofuels and fuel cells, etc. And coming for an food and nutrition applications, they have a vast range of applications like in dairy products, wines, nutrition, making and probiotics, and coming for an biotechnology applications. This they have a major role in making an synthetic antibiotics, or by using recombinant technologies, and coming for an biosensors. They help in making an enzyme estimations and assay productions, and coming for an environmental applications like biodegradation of toxic compounds, like making an xenobiotic compounds, synthesis of an xenobiotic degrading microorganisms like in superbug creations, and coming for an other industry applications like paper industry, steel, leather, uh, textile, fiber, detergents, say for like making an anti-corrosive steel products. So where the microorganism helps to have a long lasting life for the stainless steels. That's called an anti-corrosive agents, and coming for a medical industry, so helps in making and diagnostic aids and making an antibiotics, like developing a new drugs. So it's having a vast measure of major applications, which helps to make it the role of microorganisms are very essential, as they have been seen everywhere in the nature. So next, coming for the metagenomics. So of all the living organisms on the planet, microorganisms are the most numerically abundant and diverse in nature. So researchers have begun to understand their diversity profiles, metabolic functioning, and potential economic values. So in general speaking, in classical investigations, to study the microorganism, one has to culture and study the selected microbe in the lab. However, this approach can yield much information, like saying like, so if you're going to deal with a single organism, then it's simply called as of in genomics. So studying the genome of one single organism is called as in genomics, whereas if you're going to deal with a group of microorganisms, then it's called as an metagenomics. And if you're going to study the microbial community based on an analyzing the DNA within an environmental sample, then it's called as an environmental genomics. So example includes profiling microbial population in water samples taken from deep oceans, or in soil samples from human-made environments like active mineral mines, and environmental metagenomic study data are used for agriculture microbiome analysis, ecology remediation, and other biological investigations. So coming for all this, if so we need to know an RNA biology, so which 16S rRNA is very important. So as the 16S rRNA are relatively short, which will be around 1.5 kb, and these are short, and because of short sequence, the sequencing cost is very low, and these 16S rRNA are highly conserved, and definitely they are different between the species, so which makes an unique for each and every species. Though we cannot identify an species at 16S rRNA, but this is very helpful to differentiate the species, and a small piece of information, a small variable region is enough to have and much more informations. So in general, this metagenomics deals with and sequencing a group of microorganisms and sequencing the 16S rRNA of a group of microorganisms by using a specific variable regions from the 16S rRNA region. So next, coming to an 16S rRNA gene markers. So these 16S rRNA genes are generally called as often gene markers. So in prokaryotes, we have 70S ribosomes made up of two subunits of 30S and 40S, while in eukaryotes, we have 80S ribosomes made up of two subunits of 40S and 60S. So coming to in prokaryotes, the 16S ribosomal RNA is a component of 30S small subunit of the prokaryotic ribosome that binds to the Shine-Dalgarno sequence, or simply we can call as of an SD sequence. So the genes coding for that are referred to as an 16S gene and are used in reconstructing phylogenes. So it's due to the slow rate of evolution of these regions in the genes. So these ribosomal RNAs are classified into 16S, 23S, and 5S rRNA according to their sediment rates. So in general, this 16S rRNA has become standard gene in bacterial taxonomic classification because it is more easily and readily sequenced and contains enough phylogenetic information with short stretch of sequence. So this 16S rRNA gene consists of 9 variable regions, and these regions, the sequences of these regions are usually used for species annotation, phylogeny, and diversity analysis. So these are the primer sets for amplification of 16S rRNA. So there are various commercially available primers for sequencing the 16S rRNA gene. In general, the whole length of 16S rRNA gene is sequenced by using 27F and 1492 reverse primer sets, while the other primers are used to sequence by using various sequencing technologies like Sanger sequencing or PacBio Smart sequencing, because different lengths of DNA are sequenced by various sequencing technologies. So one can use the suitable set of primers for the sequencing technologies. Say for example, most commonly the people used to prefer at V3, V4 regions like 337F and 805R. So these are the most significantly used primers. Or if you're going to sequence the whole length of your 16S rRNA, then we can prefer like 27F and, as we discussed, 27F and 9 and 1492 reverse primer sets. So next, coming for these variable regions and sequencing technologies. So we have discussed that we have different kinds of variable regions like V1 to V9 regions, and we have an specific primer sets for each and every specific region. And if you want to, by looking into this picture, we can know that which region can be amplified by using which kind of a sequencing technology. Say for an example, an V1 to V3 region. So if you want to identify an V1 to V3 regions, it will be around 510 base pairs. So that can be sequenced by using Roche 454 sequencing missions. And if you're going for V3, V4 regions, will be the targeted site region will be around 428 base pairs, and this can be used for an Illumina MySeq pedant sequencing.
Again, V3, V5 regions, the targeted length will be of 548 base pairs. So can be used by sequenced by using Roche 454, and just V4 region, we can have a target region of 252 base pairs for high-seq, and V6, V9 regions is of 562 base pairs with again Roche 454. And if you want to sequence the full length of the genome, full length of the gene sequence of V1 to V9 region, so we can use an PacBio systems. So like this, one can opt for what kind of an sequencing which we are going to perform and which region we can target. And mostly it is suggested that V3, V4 regions are most specific regions to identify as number of bacteria again and archaeal cultures, isolates. So it's preferable that we can use V3, V4 regions, which suggests that most of the bacterial species can be identified by using this V3 and V4 region. So here we are going to say that rRNA gene as a marker. So we have discussed that 16S rRNA is been seen in prokaryotes and 18S rRNA is seen in eukaryotes. So these are the three domains of an life forms which has been classified into an bacteria, archaea and eukaryota. So this is an phylogenetic representation of these three kingdoms of life. So where we can identify the bacterial and archaeal species by using their 16S rRNA genes, and while eukaryotes can be identified by using their 18S rRNA sequencing. So this classification of life into three domains was first used by using an RNA sequence by Oss. So coming to the taxonomy, once we have an 16S or 18S rRNA, so we are going to find out what kind of bacteria has been present in our what kind of bacteria has been present in the source samples which we have isolated. So say for an example, once we have an 16S rRNA sequencing, we can find out to which what is the species and which genus, to which family, order, class, phylum, kingdom it belongs. So in general, the higher level of the species the higher level of the organism can be easily identified by using our 16S or 18S rRNA sequences. In general, it is said that 16S and 18S rRNA genes does not yield your species level information like what is the species, but we can arrive up to a genus level. So they say that for genus level identification, the 16S and 18S rRNA are very important. So genus level is being considered as often best. So the organism can be easily identified. Closely related species which is having an higher similarity across the 16S RNA gene can be easily identified, and typically they don't use the whole gene. So just one variable region is enough, like V3, V4 region is enough for us to say that to which kingdom or to which phylum or to which genus this specific bacteria belongs to. So I remember that once we have an sequencing of 16S rRNA, so we are going to find out to which genus this particular bacteria belongs to. And in general, they are being called as of an OTUs. That's called as an operational taxonomical unit. So at which species, at which taxonomic classification level, we have identified the specific bacteria. So we can say that, so this specific bacteria has been identified onto a family level or order or class, phylum and genus, and mostly sometimes we can say that this is at species level. So coming to the sequencing technologies which we are having, so we are going for a specific kind of a topic called as of a microbiome. So where we are going to study the whole composition of an microorganisms which has been present in our data. So this can be called as of an metagenomics or whole genome shotgun sequencing. So where we are going to find out what are the bacteria and what are the archaeas which has been seen in our samples, and fungi like an eukaryotes. So if you're going to specifically say what kind of an eukaryotes we are having, so specifically fungi. So this can be done by using an 16S rRNA sequencing and 18S rRNA sequencing. So it depends upon what kind of an experiment which we are going to do, which we are going to deal with. And So if you have been dealing with only with an bacteria and archaeal sequences, the one can go for only 16S RNA sequencing. Or if you want to find out the whole microbial community, like interaction between an eukaryotes and prokaryotes, and their helpful what's the role of them, so then one can go for an 18S RNA sequencing too. So altogether this will be called as of an microbiome. So just finding out what are the microorganisms which has been present in the whole sample set. So simply if you're going to combine the everything, then we are going to call it as of an metagenomics, or simply we can call it as of an whole genome shotgun sequencing. So this we are going to see in our later classes, like one by one. So how we are going to perform those particular steps to identify which bacterial community has been present and what's the role and all. So in general, to perform this microbiome studies or metagenomics, we we are being heavily dependent on 16S rRNA sequencing. So once we collect and samples like from different environmental sources or from any samples, so we are going to perform an 16S rRNA sequencing by using and various kinds of primers which we have discussed in the earlier slides. So say for an example, I'm going to take an V3, V4 regions or any variable regions. I'm going to sequence that 16S RNA sequencing, and then I'm going to find out what to which taxonomic classification my bacteria belongs to. So this is been called as often just a simple survey that what kind of bacteria has been present in my samples and what they are doing. So once we get an 16S RNA sequencing, just we are going to find out we are going to classify their taxonomy, and we are going to say that whether it belongs to an bacteria, an archaea, or an eukaryote. Of course, if you are going to use an 16S RNA sequencing, we will be having only an bacteria and archaea. And further, we are going to do an population analysis, like we are going to say that what's an alpha diversity and beta diversity, and which community has been overrepresented or which community has been underrepresented. So which community has been dominant and which has been playing a crucial role. So this kind of survey will help us to know that which specific community or which specific phylum is very essential for maintaining the plant's health or growing the plant in different conditions like drought conditions and all. So this is what we are going to do in our further classes, and specifically we are going to call it as of an microbiome, or we can call it as of an metagenomic study. So coming for the population analysis, this is the most important analysis from our microbiome point of view or from metagenomics point of view. So the two kinds of an analysis which we are going to perform will be an alpha diversity and beta diversity. So this alpha diversity will say the difference within the sample, and beta diversity says the difference between the samples. So here we are going to consider and two important factors like evenness and richness. So evenness says the distribution of taxa, so how the distribution of the taxa has been present, and richness how many taxas has been present, and rarefaction curves, so which says that alpha diversity versus the number of observations. So this is used to test whether an environment has been sufficiently sequenced to observe all taxa or not. So that is, it says that whether the samples has been evenly sequenced or not, so that we can have an proper statistical analysis. So whether the sequencing quality is, whether these samples can be considered or not for my further analysis. So coming to this population analysis, it's very important to identify an alpha diversity and beta diversity, and sometimes we'll go for an gamma diversity too. And remember that we need to identify and the evenness and richness of our samples. So coming for the alpha diversity is within sample diversity. So it shows how many different species or OTUs are in each sample. That's called as alpha richness, and how evenly they are distributed. That's called as an evenness. So which together we are going to call it as of an alpha diversity. So say for an example, in this we have and two communities like community 1 and community 2, so which can illustrate you and richness versus a diversity. So say for an example, the community 1 is having and same richness of 4 tree species. So where you can see that the community 1 where the A, B, and C, D trees are equal in distribution, like 25%, 25%, 25%, and 25%. But in community 2, we can see only the presence of A, tree A is higher, like 80%, and B is of 5%, C is of 5%, and D is of 10%. So this says that community 1 is more diverse than community 2. So simply saying that both both the forests have the same richness, like four tree species, but community 1 has much more even distribution of the four species, while community 2 is dominated by three species. Community 2 is dominated by and three species. Though this makes community 1 more diverse than community 2. So next, coming to the beta diversity. Beta diversity is between the sample diversity. So here it shows how different every sample is from every other sample. So in general, few beta diversity analysis are made up of and completely made up of numbers. So you can say that while calculating beta diversity, few diversity analysis calculations like Bray-Curtis and Wade unifrac will consider only the abundance of OTUs will be taken into account, whereas um, the for richness calculation in beta diversity like Jacquard and unweighted unifrac methods, the presence or absence of an species will be considered. See, in this illustration you can see that the table, so how what is the difference between the samples, like sample 1 to sample 1 0, sample 1 and sample 2 it is 0.345, sample 1 to sample 3 it is 0.194. So these numbers will say that so how different the samples are with each other. So 0 represents that there is no significant difference, and so we have to perform an statistical analysis, and we can say that whether there is any statistical significant difference between the samples or not. So these are the things which we are going to do in our further classes. And usually this table looks like very we cannot interfere many things in this. So usually this the best way to visualize this beta diversity or how different are samples from each other is by using an NMDS method. It's called as of an non-metric multi-dimensional scaling, and this method will be simple as is similar to an PCA or PCO analysis. So these are the things which we will be discussing in detail when we're going for and further classes. So now coming for the environmental metagenomics. So environmental metagenomics is the study of organisms in a microbial community based on analyzing the DNA within an environmental sample. So the examples includes like profiling and microbial population in water samples taken from deep oceans or from soils from human-made environment like industries, water samples, industries, and from marine sources. So these environmental metagenomic studies are used for agriculture microbiome analysis, ecological remediations, and other biological investigations. So in general, with the ability to combine many samples in a single sequencing run and obtain high sequence coverage per sample, NGS-based metagenomic sequences can detect very low abundance members of microbial community that may be missed or too expensive to identify using other methods. So such as most microbes present in the environment are unculturable while using currently available media or methodologies. So to focus to overcome this problem, the development of new technologies that can yield large, reliable, and robust data sets that also includes genomics, transcriptomics, and proteomics have gained an attention. So at this point, the development of high throughput sequencing technologies has dramatically advanced to conduct the analysis of microbial species diversity and their functioning within the ecosystem. So this environmental metagenomics was in field that was extremely limited prior to the advent of new generation sequencing technologies. So the NGS provided the researchers with the capacity to profile entire microbial communities from complex samples and discover new organisms and explore the dynamic nature of microbial population under changing conditions. So in this common NGS methods for environmental metagenomics studies that includes four different kinds of sequence methodologies. One is of an shotgun metagenomics, and next one is of an 16S rRNA sequencing, and next is of an microbial whole genome sequencing, and de novo assembly, and microbial meta transcriptomics. So in general, once the researchers can obtain the millions of sequence reads from the selected genome through NGS technologies, they will be having a sequence reads, and it's possible for them to reconstruct the large portion of the genome by following sequencing assembling strategies in which the overlapping sequence reads are paid much attention. So however, this assembly stuff is very challenging because the sequence reads are generally quite small and genomes often contain internally repeated regions, so which may confuse the complete reconstruction of the genome from its basic reads. So in these cases, we have different kinds of programs like advanced assembly tools, and we have de novo assembly tools. And in an advanced assembly tools, one can use and reference genome sequences and follow the methods, and one can annotate the genome. However, this genome annotation is a process of identifying the important features contained within the genome and identifying the biological information to those features. Apart all from this, in this environmental metagenomics, so one can collect the samples from various extreme environments or from and basic environments like soil, or they can study the samples from water, study the samples from human tissues and space. So once the samples has been collected, the libraries will be prepared, and these libraries will be taken for and sequencing technologies like what are the technologies which we have discussed. And then this once the sequencing technology is giving you an output of raw reads, so these raw reads will be taken for further consideration of analysis like which we have discussed in 16S RNA sequencing. Like once the sequencing has been done, we are going for a survey to find out what kind of bacteria has been present, what are the bacteria has been present, and then we are going to further do an taxonomical classification of that OTUs, and we are going to say that we are going to study the population analysis like alpha, beta analysis. So these are the various advantages of performing an environmental meta genomics. So coming to the most important sequencing technology in metagenomics, it's an 16S rRNA gene amplicon based metagenomics. So we are going to perform this to find out the taxonomic profiles and structures so that we can know what bacteria are present and what they are doing actually. So here this follows and simple basic steps like collecting the samples. So the samples can be from a complex environmental microbial community. So once the samples has been collected, so we are going to extract and purify the DNA, and this extract and purified DNA will be taken for an amplification, that's called as a library preparation. And after amplification, and we are going to perform an sequencing analysis, 16S RNA sequencing. So for an example, here we can use an V3, V4 regions primers to amplify, and then we are going to follow an MySeq sequencing technology. And once we get an sequencing reads, so those reads will be further processed like an pre-processing steps like filtering what are the poor quality regions, filtering those stuff and making and removing the chimera regions. So once it has been done, so we will be getting an OTUs. The reads will be considered as of an OTUs. So we are going to cluster that OTUs and we are going to analyze that. So that's called as of an OTU analyze and clustering. And once it has been OTU clustering and analysis has been done, we are going to refer that to an reference genome databases like for classification of this OTUs by using and reference databases like Silva, RDP, and green genes database and easy taxon database. So once we classify the OTUs, so then we are going to check for its relative abundance and to which classification it belongs. So this relative abundance will give you a clear idea that who is there present in the taxonomy. So what's the role? So which community is highly, which phylum or which kingdom, which phylum or which genus is highly abundant and which is low abundant. So that's the one way of doing an relative abundance, or by going through an OTU analysis and clustering, or directly we can say that it's an taxonomic classification by using an extract alignment tools like one can do with BLAST, and it's a tedious process where, but the simple steps like once the amplification sequencing of 16S RNA gene is been done, we can go for an OTU analysis and clustering, and from there we can go for an OTU classification by using an reference database, and then we are going for an relative abundance and to give an taxonomic classification. So this can be done with an CHIME2 pipeline, so which we're going to discuss briefly, so how the pipeline works like. And once we have an OTU analysis and clustering, we can go for functional predictions, like we can identify the potential functions of the particular community and what actually they are doing. So for this, we are going to use different kinds of tools like Picrest, TaxForFun, Wicodac. So these are the tools which has been used for predicting functions of potential microbial communities by using the genomic informations from ancestors and closest relatives. One can make an functional potential, one can predict and functions, and that can be represented as of an heat maps. So this all about the 16S rRNA pipeline, so gene amplification for metagenomes. So these are the steps which we are going to perform in our course of fourth session with an hands-on training with some simple microbiome data sets. So this is all about our taxonomic profiling. So how we are going to perform. So this is the CHIME2 pipeline, so which we are going to discuss more detail when we are going for the further lectures. So here I just want to outline you what we are going to do here, and this is the basic pipeline which we are going to use for any kind of an microbiome or metagenomic data set. So once we get an raw sequences from our NGS technologies. So usually these sequences will be given with an barcodes. So we have to demultiplex. So those are called as an multiplex sequences. So once we receive an raw sequences, we need to demultiplex it, and we need to find whether it's an single end sequencing or paired end sequencing. So if it is an paired end sequencing, join the pairs and then demultiplex the sequences, and once it has been demultiplexed, so we are going to denoise or the clustering. So this denoise is nothing, is just like removing filtering the poor quality reads and any errors and removing the chimera sequences and all. So this can be done by using two different kinds of an algorithms like DADA2 or Dibbler. So once it's been done, we are going to cluster our OTUs. So once the OTUs has been clustered, so we will be having two kinds of an outputs. One will be an future table and one will be an representative sequences. So once we get an future table, one can go for an statistical analysis, or one can plot, so saying that what is the differential abundance and bar plots or heat maps, or one can perform their analysis by diversity analysis like alpha beta diversities analysis, and then with the help of an representative sequences, one can go for an building an phylogenetic tree by using the alignment methods, by using an aligned sequences. So both the rooted trees and unrooted trees can be built by using the phylogeny option. And on another hand, the taxonomic classification can be done. So saying from kingdom to the species level. So most of the cases, it's an operation taxonomical unit will be at an genus level. So the accuracy will be more until genus level. So these are the overall pipeline for your microbiome analysis or metagenome analysis. So simply saying that once you get an raw sequences, just go for a demultiplexing. If it is a paid in, join the pairs and then make it as a demultiplexing. Then going for denoising or clustering, then we are going to make an representative sequences, and these representative sequences will be used for building and phylogeny and as well as for taxonomic classification by using different kinds of databases like RPD, Green Genes, or Silva, or Easy Taxon. So once the taxonomic classification has been done, we can make we can see the differential abundance, or we can make an bar plots or heat maps, and we can study and diversity analysis like alpha beta diversity analysis, and then we are going to plot that we can perform some statistics to know the significant difference, so by using an R statistical tools. So this all overall summary of our time to pipeline, so which we are going to study in further classes in more detail. So this is what we are going to learn in this advanced environmental microbiology. So all right students, this is all for today. So in next further coming classes, we will go more deep about each and every section of the course, and we'll try to understand so how the bioinformatics has been helpful for us to analyze the microbiome or metagenome data analysis from an environmental samples, and we'll try to understand what tools are available for us to easily and how we can use them the best. So that's all for the class today. Thank you. Thank you one and all.